All right. Good morning, everybody. It's nice to see you here, and hopefully you'll join us for the rest of the week. I think we have a lot of good soybean information for you. I'm Philip Rosebaum. I am the SDSU Extension IPM Coordinator, and I do a lot of research, a lot of field research in soybean, so insects primarily. So you'll see that tomorrow with Adam Varenhorst. But welcome to today's crop hour session, and thank you for joining us. Please feel free to ask questions at any time using the Q&A button at the bottom of your Zoom screen, or make comments in the chat, and you can do that throughout the presentation. So if you have a question, please feel free to type that question when it comes to you. At the end of today's presentation, we will display a QR code for you to obtain CCA credits if that applies to you. Keep in mind, in order to scan that code, you must download the Certified Crop Advisor app and use that, and use the scan course code function. Without that, you won't be able to get your CCA credit. So we'll display that at the very end on the screen, and please use your phone to scan that. If you have any technical difficulties, please feel free to send us a chat or raise your hand using the chat and raise hand button at the bottom of the screen. All right. So with that, we have two of our experts today talking about diseases, primary or somewhat on soybean cyst nematode, but other diseases that they see. Our two presenters are Madeline Shires, our SDSU Extension plant pathologist, and our other expert is Connie Strunk, our Extension field specialist in plant pathology. So with that, I will hand it off to you, Madeline and Connie.
Okay, I am going to go ahead and start sharing our presentation. And okay, slideshow. Okay, can everyone see the screen? I'm hoping. Okay.
Yep.
All right, so we're going to be talking a lot about SCN for the first bit of this presentation, and then we're going to talk about some other soybean diseases that we diagnose through the Plant Diagnostic Clinic. And then toward the end, we will also cover some other diseases that could pop up, especially if we have a little bit more moisture this year for our crops. So this slide here kind of talks about what the most commonly reported plant disease issue on soybeans is in the northern United States. It's pretty easy to see that soybean cyst nematode is attributed for the most losses, followed by white mold, sudden death syndrome, charcoal rot, different seedling diseases, pod and stem blight, and Phytophthora root and stem rot. All of these diseases we do see from year to year in South Dakota. So we will go ahead and get started. Soybean cyst nematode, let me make this smaller. Okay, so soybean cyst nematode, it's also called SCN, which is what many of you probably know it as. It's the most devastating soybean disease. Up to 30+% of your yield can be lost before you ever are able to see foliar diseases or foliar symptoms in your plants. There is SCN resistance developing to the PI 8788 lines, which are the ones that are typically recommended to manage SCN issues. This picture here, you can see the blue circles. Those are indicating the different cysts, and those cysts are essentially the egg sacs and hold 200 to 300 eggs each. So you can see this root has thousands of eggs contained on it. This figure here is from a publication that we put out last fall. A indicates tons and tons of those little golden cysts on the roots. B, it's kind of a closer up, so you can see just how many cysts can be stacked on a single soybean root. C, these are a zoomed in picture of individual cysts, and D is also the same.
And I just wanted to pop in and just say, with the SCN, they're very small compared to the nitrogen fixing nodules that are on the roots of the plant. So there is a considerable size difference between the two. You know, oftentimes we'll say like they're about the size of a piece of little gravel or sand. So just a very, very tiny cyst or type of a pencil lead versus like an eraser, like the nitrogen fixing nodules are about a pencil eraser, where the cysts are a much smaller in size.
Okay. Thanks for adding that, Connie. This slide here is just, we took a couple of those individual cysts off of those previous roots and actually popped them, and you can see the multitude of eggs that are coming out. These, again, the arrows are indicating individual cysts or individual nematode eggs. So here in South Dakota, we know of 34 counties that have SCN currently reported. That started in 1995. The last full survey that we did to track the spread of it across the state was in 2019. We suspect that there are probably more positive counties, and this year we'll be working to do a little bit more surveying to see if it has spread. The dangers of SCN, again, kind of as I mentioned a couple of slides ago, your yield is greatly reduced if the SCN is unknown or uncontrolled in a field. SCN does not only affect soybeans, it affects many other cover crops too. And when I say affects, I mean it can feed and reproduce on other cover crops. So crop rotation with non-host plants can be very useful to help manage SCN. Another thing that we are going to continue talking about in this presentation, and you're probably going to hear a lot more about, is that our PI 8788 lines are getting less effective. They're less resistant. SCN is able to feed and reproduce on those plants. So, and that's something we'll spend a little bit more time talking about here in a minute. Anything to add, Connie?
Nope, I was just going to say, you know, with weeds, so some of the weeds can also be hosts, such as the dead nettle and others.
Yes, true. So now we're going to cover a few ways that I've kind of been mentioning to try to manage SCN. You can do that by planting those resistant cultivars. One of the things that they are recommending at the national level is rotating those different resistance genes. So that's one thing to think about. Crop rotation, especially with a non-host crop such as corn, could be very effective. Also, again, rotating that source of resistance, because we are seeing nematodes being able to reproduce on those what should have been resistant plants. Nematicide seed treatments can be very effective. And as Connie mentioned, weed management is also something to consider. If you have high SCN numbers, as like cover crops, SCN can also feed on many weeds and continue multiplying. I have a video. Give me just a second and I will pull it up. Okay. We have a video to talk about the SCN Coalition. If you haven't heard of them before, they are the kind of the national level for managing SCN. And so I'm going to play this little video that they've made. Stop. Okay. Going back to the PowerPoint now. Okay. We will start here. There we go. So hopefully you kind of saw in that video what I was talking about, where they're starting to see the resistance of those different lines not being as great as it could have been. So one of the things that we are promoting again this year is, as of right now, we do free SCN testing and HD type testing if you meet the minimum level for South Dakota growers that is funded through the South Dakota Soybean Research and Promotion Council grant. We do that in the plant diagnostic clinic. So why should you be SCN testing? It helps us monitor the movement of SCN across South Dakota. It does help us track HG types, and especially our levels of being able to do that, we need to have 10,000 eggs per 100 cc's of soil. And it can also help you and others make informed management plans to help manage SCN numbers. So I talked to our diagnostician, Connie Tandy, and got a few stats from her for 2022. In 2022, we had over 300 SCN tests submitted by South Dakota growers. Most of those did have SCN at some level. The average count for 2022 was 2,000 to 3,000 eggs per 100 cc's of soil. We had less than 10 samples that met that HG type testing at 10,000 eggs per 100 cc's of soil. But we did have a few of those samples come in. The general management recommendations: if we at the diagnostic clinic find fewer than 500 eggs per that soil sample amount, we recommend that you start thinking about it and just, knowing that you do have SCN in your field. If we see 5,000 eggs or less, we will be recommending that you really start using a management plan and resample just to make sure that your numbers were accurate. And if we start seeing 10,000 or more, where we will definitely be recommending that you have a management plan considering crop rotation. Definitely try to use resistant crops or resistant genes in your soybean crops. So that's just so you kind of understand our general management recommendations. Connie, do you have anything to add on any of these?
No, I think that's good.
Okay. Again, SCN Coalition, they've got so much information out there. If you have any questions, feel free to reach out to me or Connie, or check out their website. It's very informative. And again, kind of from that video, they again recommend that you, they recommend, and we are recommending here in South Dakota, that you test your fields to know your SCN numbers. Even if you have tested fields 6 or more years ago, it would be a good idea to test again to see if your numbers have increased or decreased. If you have never tested for SCN, you are in the eastern to middle part of South Dakota, it would be a great idea to test just to see if you have SCN problems in your field. And then again, like we've already mentioned, rotate the resistant varieties, rotate to non-host crops if your numbers are very high. And there's also some great nematicide seed treatments out there that could be utilized if needed.
And just real quick with testing, feel free to target low-lying areas, the fence rows, field entrances, maybe areas in the field that have had a lower yield drag. If you see on your monitor, test them separately from the rest of the field. You can, grid sample the entire field. So there's a couple of different ways to go about sampling just to kind of check and see within your area. So encourage you to do that. And ultimately, as you go through each, whether you're doing treatment areas of low-lying areas or, field, ultimately the test sample is about a pint of soil that you'll be submitted into the diagnostic clinic for analysis. So it's not that much. You'll take a lot of, soil cores. And if you don't have a soil probe, feel free to use a spade to dig those samples. You're about 0 to 6 inches depth is what we're looking at, right in or as close to where soybean roots would have been in the field. Mix those cores or, shovelfuls of soil together to get about that pint that you'd ultimately send in.
Yep, and I'll just add on to that if you are wanting to sample for SCN and need help or recommendations, feel free to reach out to Connie, me, or to the plant diagnostic clinic. We can provide as much information as you need about getting good samples for SCN. Okay, so now we are kind of done with the SCN portion of this talk. I am going to start into some of these other diseases that we do see from year to year. The first one of those is charcoal rot. We do see this at low levels most every year. This is a severe infection in, I believe it was Indiana from a couple of years ago. Whenever we do the diagnostics for charcoal rot, we split the soybean stems and look for these little black spots growing within the stems. That's actually the fungus that causes charcoal rot. The little black spots are the fruiting bodies that produce more of the fungus. Anything to add, Connie?
Nope.
Okay. So some symptoms of charcoal rot. Again, it is a fungal disease caused by Macrophamina. I can't say that last name right now. Warm summers, warm winters, and drought favor development of charcoal rot. The fungus does overwinter in the soil. So if you have observed charcoal rot in your field one year, you're very likely to see it again next year, especially if you're growing soybeans back to back. The fungus infects the plant through the roots. It begins infecting the plant very slowly until pod fill is complete. After pod fill is complete, it begins rapid reproduction, which causes plant collapse. And that's because it's in the stem of the soybean and reproducing there. This fungus can infect the seed of soybeans, seedlings, as well as mature plants. This is the disease cycle for charcoal rot. So we start here. You can see that the fungus survives in the soil or on soybean residues. Once the new year's soybean crops come in contact with those surviving fungal structures, it begins infection in the roots. After it has established infection in the roots, it begins growing in the stems and the roots, which affects the way that the plant is able to take up water and nutrients. And again, once it kind of hits that pod fill life stage, the plant it begins reproducing a lot and completely clogs the nutrient transport system, the phloem and vascular tissues. And you end up with the stems having that charcoal looking appearance because there's so much fungus growing within the stem. And after that, it starts over again as those residues end up either staying in the field or getting tilled into the soil. Here's some other pictures of charcoal rot. This is the one we'll see many times if it's a heavy infection when it comes into the diagnostic clinic. This gray discoloration can be an early sign that there's charcoal rot developing and preventing the flow of nutrients. This is actually the charcoal rot fungus growing on the outside of the stem. You can kind of see those here. Okay, anything to add, Connie?
Nope, you're doing great.
Okay. The next one that we're going to talk about is Phytophthora root rot of soybeans. We do have this here in the state. We usually get several samples of it into the diagnostic clinic, especially during years with more rainfall. It can be very devastating to a field whenever this disease is established in that field. There are different management strategies for Phytophthora. There are resistant cultivars. There's several different resistance genes out there that can be used. One of the best ways to help manage Phytophthora is to improve drainage of your field. The Phytophthora spores that cause infection move with water, so improving that drainage does help. And then there's also some seed treatments, as Phytophthora typically causes infection early in the year, especially on the soybean seedlings. Moving into, oh, go on,
I was
Connie.
just going to back up real quick with just the symptom of Phytophthora. Again, a great, with Phytophthora, it can take out seedlings and it can take out fully mature plants. So it's a disease that we can have, take out plants the entire season long. So as you see, a lot of seedlings here have succumbed to this disease, but we can also find older plants that have Phytophthora. A good symptom or rule of thumb when you're scouting and taking a look is you'll see a chocolate brown lesion that extends or starts at the soil line and it moves up the soybean stem. And so that would be typically what you're looking at for Phytophthora. And again, it can attack and take out the seedlings from a very, just emerged stage or before up to, like a fully mature plant. So we can be losing plants due to Phytophthora throughout that entire growing season.
Okay, thank you for adding that. The next disease is sudden death syndrome. We did see a little bit of this in South Dakota last fall, but not too much. The symptoms of it are the green veins and the blue mycelia, which you can kind of see in this picture here. Here's some other pictures from, I believe it was Minnesota of SDS. Again, you can see those green veins that remain on the leaf and a little bit of that blue mycelia that's starting to develop in here. Sudden death syndrome is also a fungal pathogen. Most of our soybean diseases are caused by a fungus. This SDS is caused by Fusarium species. The infection typically happens early in the season, especially if the crops are planted in cool, wet soils early in the planting season. Symptoms of SDS typically do not appear until July or August. The fungus is soil-borne like many of our others, and it has been shown to affect some other bean crops. This here is another life cycle example of how the fungus causes infection. So it starts here and it can survive in the soil and on soybean and corn residues. It infects the soybean plant early in the season. It then starts growing on root tissues and produces toxins in the roots that eventually start showing developing those foliar symptoms. After that has occurred, that's when the foliar symptoms begin to develop, and the leaves will typically drop, but the petioles will usually remain attached to the stem. And at the end of the season, after harvest, the inoculum is again able to stay in the soybean and corn residues to overwinter and start the infection cycle again.
Yeah,
there are, oh, go ahead, Connie.
Well, I was just going to say, sudden death is a type of disease that affects the plant or the seedling, after planting very early, enters into that seedling, but then it kind of lays wait as it's kind of going through its life cycle, waiting for the flowering time frame. Oftentimes we'll see it after we reach our reproductive phases, is where it wants to kind of rear its ugly head. So where the name sudden death is, for some folks, as you're out scouting, the fields look fine, and then all of a sudden, you're close to flowering or you're just starting potties, then all of a sudden things start to change, and you're gonna, you start to see very suddenly the leaves having those green veins, the yellow, the necrosis. But the nice thing with this disease is it doesn't take out the entire field. It's usually pretty isolated within the field or within the areas. So again, this is a type of disease that, affects early. You don't know you have a problem until once we've reached those reproductive phases.
Okay, so there are some different ways to manage SDS. There have been shown to be some tolerant cultivars because the fungus does kind of prefer cooler, moisture soils. Planting in warm, well-drained soils could be an option. There are also some fungicide seed treatments out there if that could be utilized if SDS has been a problem in the past for that field. Now I'm just going to briefly mention some other fungicide timings for white mold management, which we'll talk about in a minute. Those should be applied at R1, which is the beginning of flowering. And that's because there's specific active ingredients within that fungicide that are only really effective at R1. For many of these other fungal diseases, the best fungicide timing is R3 to R4. And again, we'll be talking about some of those in the next few minutes. Frogeye leaf spot is another disease that we do see here in South Dakota, and the added challenge of it is resistant to some of the different fungicide classes that we have available. Frogeye leaf spot, if you have not seen it before, it starts as circular to angular lesions on the leaf. They are dark brown spots with a reddish margin, so you can kind of see that on some of these. As the fungus gets more developed on the plant, the lesions become an ashy gray color, and they may eventually form or merge to form these much bigger lesions on the leaves. And that can lead to defoliation if the lesions get big enough. There are some different ways to manage frogeye leaf spot if it is a problem in your field. There are varieties with some resistance or tolerance to FLS. You can also practice crop rotation and tillage when practical to reduce the inoculum loads. A foliar fungicide can be applied between R2 and R5 when the disease pressure is likely to be high. Another thing, because this disease does have resistance to fungicides, make sure that you alternate the fungicide chemistries if you're having to manage frogeye leaf spot. Anything to add on FLS, Connie?
I was just gonna say that the timing, so the R2 would be the full flower, the R5 would be the beginning seed, just to explain what those reproductive phases are.
Okay.
And then with frogeye, sometimes, it can look like an adjuvant burn or something. So it's really, the big thing with any of these diseases, if in doubt, send in samples, send in pictures, get it properly identified to make sure that you are working with, in fact, a disease or a fungal disease, or it's really important knowing what you have out there.
Yes, and if you are needing to figure out where to send things into, don't forget about the plant diagnostic clinic because we can definitely help you out. All right, moving into another disease, which is white mold that we mentioned earlier. White mold it pops up some years in South Dakota and it's really bad. Other years, especially the drought years, it's barely seen. In fields where it's very established and causing problems, you can see some yield loss. As the fungus starts infecting the plant, it starts reproducing within the stem and forming these little fruiting bodies here. And this is kind of a close-up on those. This is kind of the life cycle. Again, like so many of our others, it starts in the soil, and then it is able to start reproducing once it finds a plant to grow on, and it causes infection because it blocks the vascular system in that plant stem. And you can kind of see here, this is kind of a real world example. This is a very advanced infection, and the plants are declining quickly, and our yield is greatly affected. Again, there are some tolerant cultivars. Sorry, there are some tolerant cultivars. There are some fungicides, especially that can, if you use them at the R1 time point. There are also some biological control options out there. Crop rotation can help manage this. So yeah. Then this is that next slide that popped up. This is just an example of a study that was done looking for timings to treat white mold. The non-treated and R3, you can see the severity is very much the same, but R1, it was lowered. So this is just a great way to show that the timing for fungicides is extremely important.
And why this fungicide timing is so important is the white mold disease infects during flowering. So if conditions are right, we're having some moisture, that disease pressure, spores are in that air, it'll enter during that flowering time frame. So that's what that R1 is, those beginning flowering. If we wait to treat or put on a fungicide for white mold control at the R3, which is that beginning pod stage of growth, we've missed that window, right? All of the flowers would have formed. So we want to put on the fungicide as a preventative at that R1 or beginning flower time frame, because that's, again, when this white mold disease infects here. And that's when how that disease replicates and works through. In South Dakota, we tend to see more of our hotspot of white mold up in the Brown County area, but it is a disease that we can get anywhere throughout the state. Again, just more of those conditions and some more of that moisture.
Okay, so the next disease is pod and stem blight of soybeans. Again, this is another fungal disease. The symptoms are small black fruiting bodies, which are called pycnidia, that grow on the outside of the stems and the pods. And this picture here I thought was a great example of that because you can see those very clearly. This fungus, like so many others, survives in the residues, and it can also survive on seeds. It is able to infect new plants through water splashing because the fruiting bodies that hold that next generation are on the outside of the infected plant. These are some other pictures of pod and stem blight. You can see pretty clearly here the problems that it can cause. And this is another diagnostic that's used by splitting the stem and looking to see what it's looking like. Anything to add on pod and stem blight, Connie?
Nope.
Okay, so moving into stem canker. This is another fungus, another fungal disease. It's from the same genus as the pod and stem blight. Dead plants with dried leaf, the way that you're going to know that you're having stem canker is you'll have dead plants with dried leaves remaining attached on the petioles late in the season. Stem canker typically occurs in patches in the soybean field. The lesions start as small reddish brown spots at the base of the branch or the leaf petiole. They then expand to form sunken cankers that are reddish brown with reddish margins. The cankers can be on one side of the stem, and they can also extend over several nodes or girdle the stem, killing the plant. It's very common for those cankers to cause top dieback by forming a dark brown canker in the upper 4 to 6 internodes when killing only the top of the plant. A diagnostic symptom is gray streaking in the lower stem and taproots. Then another symptom that could be seen from stem canker is interveinal foliar chlorosis and necrosis, because this fungus, like some of the others, produces a toxin. So it produces some of those weird yellow green symptoms that you'll see in the leaves from that. So this is an example of stem canker, and you can see that forming here. Another close-up. Some more pictures. So you can see it forming here on a node from the soybean. And then this is a more advanced stem canker. And another little life cycle. The fungus overwinters in the crop residue and soils like so many others. The rain splashing moves the spores from that plant residue and soil onto new plants. The lesions of stem canker are typically going to show up during the reproductive growth stages, and they will continue to develop and get worse and can girdle the stem, leading to wilting or plant death, or again, it could just kill the top part of the plant. It depends on how advanced that disease is able to get. Anything to add on stem canker, Connie?
Just, for that diagnostic sampling, again, if you see a lesion or that spot where the soybean leaf attaches to the stem, or with the leaf, at that petiole or at the base of the branch, if you will, that's where you're going to see that canker start. It can be, very small, it can be large, it can be on just one part of the stem, but oftentimes it'll go around or kind of girdle, like it'll continue to grow around that stem. And it can just be on the, the one node or one petal area, or it can stretch and go over multiple, internodes or nodes. So it can grow to be quite big, but it can also be very small.
Okay. So the next few diseases are some that we see less frequently here in South Dakota, but they do pop up, especially if we have a wetter year. The first one is bacterial blight. The symptoms of that are small, angular, reddish-brown lesions surrounded by a yellow halo, and those are going to be found on the leaf in this example photo here. The lesions can grow together to produce large, irregularly shaped dead areas, which you can see on both of these pictures. The centers of the older lesions frequently fall out, which causes this tattered appearance. But the one nice thing about bacterial blight is that it very seldom causes yield loss, but it can be problematic and a little bit worrying to see the leaves not looking so great on your crop.
It's very common. Pretty much every South Dakota soybean field in a typical non-drought year will have some form of bacterial blight out there.
And while this is a bacterial disease, it acts very much like the fungal diseases. So it overwinters in the soybean residues and seed. The rain or watering that occurs can splash the bacteria onto developing plants. The leaf lesions that we saw in the previous pictures, they form early in the season and are able to actually produce more bacteria inoculum to continue the infection. As the season continues on, the lesions grow together and cause leaf tattering. Cool, wet weather is favored for disease development, and then the cycle starts over again. Anything else to add, Connie?
No.
Okay. Then the next one is bacterial pustule. And this is, again, a bacterial disease. It's caused by Xanthomonas species. It is a mid to late season disease that occurs when temperatures are warm. The symptoms start as small pale green spots with elevated centers and develop into large irregularly shaped areas with yellow halos, and you can see that pretty clearly happening here. Pustules may form in the lesions on the underside of the leaf, and I have a picture of that on the next slide. The infected leaves will not tatter like bacterial blight. The disease development is favored by hot, wet weather, and there are, it's recommended that crop rotation and tillage be used to reduce the survival of the bacteria. And so these are some more examples. This here is the pustule that they were talking about that grows on the underside of the leaf. And you can see that even when the disease is pretty bad on the leaves, it doesn't really cause those tattered edges or margins like the other bacterial disease did. Anything else to add, Connie, before we start on this next group?
Just with bacterial pustule, we do see it, you know, again, with more of that wet and warm type weather. We don't have to worry about it, but it is a disease that can be easily misidentified for like Asian soybean rust. But good news for us, we don't have the Asian soybean rust, so that helps out. So just, you know, like I said, get things sampled and identified.
Yes. Okay, so we're now going to talk about a couple of viruses on soybeans. Connie, would you like to talk through these since you've got a little bit more experience with those than I do?
I certainly can start with,
Okay.
with our soybean viruses. There's two kind of like the main diseases that we, actually three, and I think we only show two, but anyhow, that it's okay. With soybean diseases, we can find alfalfa mosaic on soybean leaves, a lot of with our viruses. You know, if it's we're showing our pictures of our, what we call our textbook samples, meaning they're really showing what we're wanting to show you, which is characteristic of this disease. So a lot of times with our viruses, you're going to see different color variations. So you're going to see some yellowing and some either lightening of the green or darkening of green, but you're going to see like the two-tone kind of appearance. You're going to see some, we call it distortion or rugosity. So it looks like the leaves are bumpy in nature. The problem with viruses, no matter what crop we work with, they will give the illusion that it could be, let's say, a herbicide drift, or they could have been tainted with something. So that's where we, you know, can't stress enough that properly getting things identified. Generally with our viruses, you're going to see the highest symptoms and experience more of the yield loss if they start out earlier. So your youngest tissue is always going to be the most, showing the most symptoms. As the plants age or those leaves age, you're going to lose some of that color distortion. You're just going to, you know, lose a little bit of it. So you can't just go with looks alone. You want to go ahead and click the slide. With alfalfa mosaic, it is, I think we labeled this one wrong, but anyhow, bright yellow, dark green tissue, newly emerged leaves. They may be small with bright yellow spots and brown discoloration. Oftentimes with viruses, plants may be stunted. With soybean mosaic virus, looks a lot, you know, looks the same with some of that discoloration. Anyhow, it's transmitted by more than 15 different aphid species. Primarily here in South Dakota, it is the soybean aphid. The alternate host for can be with viruses can be on alfalfa, other legumes, and then tends to be on that nightshade family of weeds is where you could find the virus here. We'll go ahead and click through here, looking at some of the different symptoms and pictures of what viruses can look like out in the field, right? So we've got a lot of green tissue on that top left one. We'll have some leaves that are really showing that color discoloration. So it's not something with viruses, you know, like any disease, you might have it on really showing the symptoms on one or two petioles or stems, or it may take out the entire plant. Really, again, it depends on when that infection moves through. You know, you might have more of a darker, just more of looking at some of the veins. You might have more spotted or splotchy in nature on the leaves, as you kind of see on the picture on the third one here. The fourth one, you're really starting to see that little bit darker, older looking of the green, the yellow where it's just not as, we'll say, really golden pronounced, you know, a little bit aged of a yellow. You're starting to see that distortion, like kind of maybe changing the shape of the leaves a little bit, some of the bumps. On the bottom left here, you know, looking at the two rows that are kind of in the forefront of that picture, they're more yellowed in nature. So they're virus compared to, let's say, non-virus infected plants on the surrounding or the background. They're a little bit stunted, again, showing some more of that rugosity. So, you know, colors alone aren't necessarily indicators of virus, because what happened or what do we see? Well, oftentimes with SCN, we have stunted plants and we have some yellowing occur. And so with that, we need to eliminate, is it SCN or is it virus? And then also we have like iron chlorosis that can cause the yellowing of plants. So we want to really, pull those samples, get them identified, go ahead and click forward. The other disease that has been an issue here in South Dakota, which is bean pod mottle virus, really was a problem, about 20 years ago, actually, when we first really started having an aphid issue and didn't really have the genetics up to play as they are now. And then also like the seed treatment or the importance of doing some of those insecticide controls early on. So with bean pod mottle virus, it's vectored by the bean leaf beetle, which is a very docile insect, multiple colors. It's just got like a little V shape behind the head, if you will. Anyhow, with bean pod mottle, again, it causes distorted leaves, often wrinkled. They have that mottled appearance, so maybe not quite as drastic as some of the yellowing, all depending, because each different, cultivar may display some of the colored symptoms a little bit different. With viruses, generally cooler temperatures have more of those pronounced symptoms. So you're going to see more of those coloration. You're going to see more of that color disparity in cooler temperatures. When seasons warm up, you're going to see some of those colors go away or kind of fade out. They're just not as pronounced. With viruses, the heat really will mask a lot of things out there for symptoms. So with BPMV, infection can decrease the number of pods on a plant, reduces that seed size, weight, and number, and it also will cause some mottling of that hilum. So you'll see instead of that nice little intact hilum, it's going to kind of spread and be dark brown, black, all depending on the cultivar and how it responds. Again, management for BPMV is done by using the insecticide early on in fields, especially if you've had a history of BPMV or the bean leaf beetles. If you're seeing high presence out there is when we want to look at controlling them, but we'll learn more about insect control tomorrow. So go ahead and click. And then again, these are just some different grouping of pictures showing BPMV out there. With, again, the viruses, you're going to have that mottled green-yellow appearance. You're going to have some of that rugosity or the distortion and bumpiness. So you're going to start to see where the leaves just aren't as smooth. Some might be very small. Some might be twisted up. Again, depends on how the level of virus is in the plant. Again, our youngest tissue will always have the most pronounced symptoms and be affected the most. The top middle picture here with the fingers and the seed really show that bleeding of the hilum or discoloration of the hilum due to this disease. A perfectly healthy soybean would not have all of that brown or black spreading down and kind of going around looking like a saddle on that soybean. We want just that little bit of hilum right up at the top. Again, with BPMV, you're going to see symptoms not only on the leaves, but you can see it on the pods as well. So again, you may limit, instead of being 3 or 4 seeds per pod, you might be 1 or 2 due to the virus alone, or you may have just a very small seed if it's trying to fill for 3. So again, diagnostic clinic, they have many different tests to see which virus you're working with. Unfortunately, with the virus, you're not going to be able to apply a fungicide or anything out during the growing season. It's kind of managing beforehand. So looking at, your seed selection and looking at controlling the vectors. Go ahead. And so we're just going to kind of wrap up here with just saying disease management. It really starts with you, really knowing your field history for diseases, scouting and keeping really good notes of what you're finding out there. If in doubt, take a picture, send it in. It's a very small investment compared to, the cost of fungicide or an improper application out there. So some of that goes in handy with knowing what is your variety hybrid disease package, doing the work now to see which diseases you would like to try to prevent or reduce out in the field. does volumes as you're going through that growing season. Really keep track of the weather, because some of our diseases are dependent on the weather and then certain growth stages. And then just want to say, if you're going to do a fungicide, so if you're going to apply a fungicide, really take into account, the timing. So again, white mold needs that R1 growth stage, where other diseases that are fungal in nature can be at that R3 to 4 is where we're really kind of pushing that timing. Look at the mode of action. Do you use just one? Could you do a mixed product? Rotate your mode of actions. We don't want to stay with the same type of one because we're going to develop resistance. And then after application, continue to scout to make sure that your treatment is working, and take notes of any efficacy or resistance issues. Because if you suspect something, send it in and we can look at that, because we do have frogeye leaf spot that has resistance to our Strobelian family. So again, we want to know that it's out there and prevent that. Madeline, I'll shoot it back to you.
Okay. Maybe. Okay. So a couple of our required slides. Here's the different resources that we used. And then here is my contact information and Connie's contact information. And with that, we are done.